as we finished last week, we figured out the way proteins are synthesized in a cell, right? And pretty much what we figured out about prokaryotic cell is that you have transcription and translation happening at the same time, pretty much, right? So you have mRNA being synthesized and at the same time, while transcription isn't finished yet. That's what we talked about right here. You already, well, your cell already starts to produce protein. This is one of the profound differences between the pro and the eukaryotic cells, because in eukaryotes, just to remind you, there's not an opportunity for such thing, right? What prevents eukaryotes from such a continuous gene expression? Why there is no possibility in eukaryotes to do transcription, to perform transcription and translation at the same time? The nuclear membrane, right, that separates, compartmentalizes the synthesis, right? There's also something else that we talked about. Yeah, so mRNA has to be transported, and there's something else about mRNA that is present in eukaryotes and absent in prokaryotes. Splicing, yeah. Post-translational modifications. Post-transcriptional modifications of mRNA. Now this is a summary of the differences in gene expression between the pro and eukaryotes. So if you would look, I tried to highlight eukaryotes in red and prokaryotes in black here. You see that in prokaryotes, what we have discussed already, the translation follows the transcription immediately. I mean, we can't even, we may say it doesn't follow, they happen at the same time. Translation occurs with a little bit of a delay, right? Does that make sense? In eukaryotes, transcription and translation are strictly separated in space and in time. Transcription happens in the nucleus, while translation happens in the endoplasmic reticulum or cytoplasm. Does that make sense? In prokaryotes, mRNA, when it is produced as the result of transcription, in the end of transcription, it is ready to get translated. There is no processing of mRNA. It's ready to go. You can see it right here in this illustration. In eukaryotes, there's processing of mRNA. You see, that's the primary transcript, and that's the transcript after the processing. Okay. We didn't talk much about processing. Out of all those processes, we focused mostly on splicing. But there are a couple more things that happen to eukaryotic mRNA between transcription and translation. Just want to point your attention to them. One, the addition of the poly-A tail, the marker for readiness. That's the transport sequence. Okay. And another, adding RNA cap structure. Well, cap in this case means what we mean when we say cap. Okay, something that gets in the head. Well, in this case, there is a special little bunch of organic molecules that sit on there. Does anyone remember what's the structure of 5' cap on eukaryotic mRNA. Something about translation. Go back one slide. Does anyone remember? What's the function of 5' cap on eukaryotic mRNA? Something recognizes it. Yep, the small subunit of the ribosome. Okay, without the cap, ribosome will not bind mRNA and translation will not start. Does that make sense? Another difference that, again, you just have to understand what the word means. mRNA in eukaryotes is monocistronic, while in prokaryotes it's polycistronic. We'll try to explain this concept, which sounds really complicated, but it is not. Monocistronic mRNA means that when transcription happens, only one gene gets transcribed. Does that make sense? Only one gene, not 2, not 3, not 53, but one gene. So this mRNA here encodes one gene strictly. Transcription in prokaryotes is polycistronic, which means that in one transcript, one mRNA, cell may have several genes. Does that make sense? Imagine that you're making copies. One case, you make a copy of each chapter of the book separately. Make a copy of the chapter, put it away, and staple it, and that's one chapter. That's monocistronic. In another case, you make copies of multiple chapters and staple them all together. Does that make sense? The difference between monocistronic and polycistronic. Again, in monocistronic, there's one gene per mRNA molecule. In polycistronic, one mRNA molecule contains several genes. Does that make sense? So during translation, those genes are translated separately. But other than that, mRNA may contain several genes. And finally, the difference between ribosomal structures. We've talked about it on several occasions. Ribosome in eukaryotes is very much different from the ribosome in prokaryotes. Not only this, so once protein is synthesized, are they ready to go? Are they ready to do things that they're supposed to do? In prokaryotes, mostly yes. Most of the proteins that are made in bacteria, for example, they're ready to go, ready to work, ready to perform whatever function it is. In eukaryotes, this protein may not necessarily be ready yet. It has often to undergo so-called post-translational modifications. For instance, chemical modification, adding different residues like carboxyl residue or acetyl residue or something else, or phosphate, okay? Because this will ensure that the protein will be functional. In some cases, newly synthesized proteins have to be cut. So it's a bigger one, it has to be cleaved. Sometimes even large protein molecules, like the protein called sumo or ubiquitin, are added to the newly synthesized proteins. All those post-translational modifications, here's the phrase, post-translational modifications, mostly happen in eukaryotic cells. And that's, but I want to say mostly. In prokaryotes, post-translational modifications are less frequent. This is absolutely mind-boggling picture, which as mind-boggling as any flowchart until you start to follow it through. Okay, so this is more of a review slide for you, and it's a great way to spell out the processes of gene expression in pro and eukaryotic cells. So here on top, well, I kind of leave it here as well. Here on top is the expression of genes in prokaryotes. So you can see a gene, okay? Sometimes gene is preceded by certain regulatory sequence. This gene is transcribed into mRNA. mRNA is translated into proteins. Okay? That's fairly simple. That's prokaryotes. That's the central dogma of molecular biology as we learn it first time. If you look at eukaryotic ones, the difference is that the gene consists of both introns, which are labeled by beige sign here, and exons. Okay, so after the transcription, primary transcript is processed into final, ready to translate mRNA molecule. That ready to translate processed mRNA molecule produces protein in the process of translation, and the protein is then modified. Different types of chemicals can be added to the protein molecules. You don't have to know them. You have to understand the concept of post-translational modification rather than know the structure of the I don't know, the acetyl group or carboxyl group. Okay. Does that make sense? This we're going to disregard so far because we didn't talk about replication. Okay, just a part of the thing. That makes sense? So this is, again, I suggest take this picture and trace it down, try to rewrite it yourself, okay, to make sure that you understand the sequence of steps. I will ask you about every step in pro and eukaryotes, okay? As I said, I never ask beyond what was in the lecture, but I may ask pretty much anything that relates to the gene expression. Any questions on that scheme? Yes. Yes, let me actually write it. That's a good point. So that's going to be prokaryotes, and that's going to be eukaryotes. So the question is the difference. So major difference between pro and eukaryotes. In prokaryotes, there are no introns and exons. Okay, there are no introns and exons. The entire gene is coding sequence. Therefore, in prokaryotes, there is no splicing. Okay, which is present in eukaryotes. And for prokaryotes, the post-translational modification is very uncharacteristic. It can happen, but it's very infrequent. Now, if you see the answer, like one of the answers to whatever question is, post-translational modification happens strictly in eukaryotes and never happens in prokaryotes, that's wrong. Does that make sense? But if I say mostly in eukaryotes and infrequently in prokaryotes, that's correct. Does that make sense? Did I answer? Awesome. Replication. Horror. Pure distilled horror. The problem with replication is that it's kind of awkward how it happens, and I would say it's kind of unnatural. I will do my best to guide you through this. I don't want you to get scared. I will try to play myself, okay? We don't have a lot of opportunities to make those little sketches, but I will try my best, okay? So first of all, what is replication of the DNA? Can anyone tell me? What's replication? Reproducing the entire thing. The entire DNA. Does that make... You see the difference? When we express the genes, do we express all the genes that we have in the genome? No. When we replicate, do we replicate everything? When we replicate for cell division, do we replicate everything? Think about it. For the next cell to be produced, yes. We copy the entire genome. During replication, when we copied, are there any proteins made? No. We're just copying it. When you copy the entire book, you don't read it. You just copy it. Does that make sense? Okay. Basically, what is... So how replication works in a very, very simplified manner. So your DNA molecule exists in which form? Double strand or single strand? Double strand. So that's your double strand. Where are the bases? Between my hands, right? Those are two strands. So in order to copy them, what I have to do with the bases? I have to split them. I have to expose the bases. Double-stranded DNA is unzipped, and each strand is copied. Does that make sense? So each strand of DNA possesses the entire genetic information, the entire genome of the organism. We have two complementary copies, like positive and negative, of the same picture. Does that make sense? Why two strands? Huh? Yes, it's pretty much a backup. Because if there is a mutation in one strand, sorry about that, I had some problems with recordings last week, so I constantly check if the mic works. If we have a problem, a break, a mutation in one strand, it can be repaired because another strand remains intact. Does that make sense? Kind of running ahead of the train, but if bases are changed in one strand, what's going to happen with the double strand structure in this part of the DNA? Like you had something like this, and then this one is changed for something else. What's going to happen there with those two strands? What you going to lose? Well, what's going to happen? What you're going to lose? You're going to lose something there. No, base is going to be different. And if it's different, what's not going to happen? Before that. Huh? Yes, it's not going to bond together. Does that make sense? So if you have something else instead of C here, not C, A, T, G, doesn't matter. Pair is not existing anymore. Does that make sense? So it's like in the zipper, you're going to have two links separated, like a little bubble. There are proteins that can recognize it and correct it. Does that make sense? But the second chain is fine. The problem is when you have the break in two strands. And we're going to talk about that a little bit, but that's the problem. Okay? Now, replication. Replication in prokaryotes happens in the nucleoid. So technically in a cytoplasm, because there's no membrane, right? So there's no strict clear distinction between like there is one existing in eukaryotes. In eukaryotes you have definitively divided nucleus and cytoplasm. So in eukaryotes replication happens strictly in the nucleus. Therefore, double-stranded DNA, and in fact, single-stranded DNA, are abnormal when they are in the cytoplasm. Does that make sense? Right? So when you have DS, double-stranded DNA, single-stranded DNA in cytoplasm, problem. Does that make sense? Now, just to remind you, I think we talked about it. There's one more structure. Is there anybody? No? Okay. There's one more structure, another RNA molecule that is abnormal for the cell. It's double-stranded molecule. Any ideas? Kind of give you a clue. It's double-stranded and it's not in this list. No. The double-stranded what? You have double-stranded DNA and single-stranded DNA covered. So double-stranded RNA then, yes. Remember when we discussed the structure of those molecules, RNA is exclusively single-stranded. So if there is double-stranded RNA, it is also a problem if it's in the cytoplasm. Well, guess what? Turns out that also all those three molecules can be found in viruses as the virus genomes. So when there's a virus infection, cell has special mechanisms to respond to the presence of those molecules in a cytoplasm. Does that make sense? Okay, enough with the digression. Let's go back to the replication. On this scheme, this is kind of a basic idea of replication. So you have double-stranded molecule, right? It gets unzipped by the enzyme called helicase. And then each strand is being copied. Okay? And remember, strands are anti-parallel. So they being copied in the opposite directions. Does that make sense? Now let's see how it works. To understand how it works, we have to figure out what are the enzymes that do it. They aren't in a very good order, but okay, first, the helicase. This one unzips two strands of DNA. Does that make sense? Next one is primase. Special enzyme that makes RNA primers. Short fragments of RNA that have to be present to start replication. They prime the process. Okay? Single-stranded binding proteins, SSBPs, also called SSBPs. When you have two strands of DNA unzipped, they actually kind of want to go back together, okay, to stick back together. To prevent the single-strand binding proteins stabilize that fork. Does that make sense so far? It's an overview. We're going to go back and look at each and every individual enzyme separately, okay? It will make way more sense. So-called sliding clamp, okay? It's a special protein structure that helps to keep the enzyme that actually synthesizes the DNA molecule so it doesn't fall off. Now this is the enzyme that actually makes the new DNA strand, so-called DNA polymerase 3, right? Now remember we mentioned primers, okay, RNA primers. Well, they have to be removed. DNA polymerase 1 removes the primers and replaces them with the DNA. Now, in the newly synthesized strain, there are going to be breaks. Those breaks have to be stitched together. That's the function, bless you, of the enzyme called ligase. It's ligates the ends. And finally, two topoisomerases. Topoisomerase 2, or DNA gyrase, prevents supercoiling of DNA. Supercoiling sounds really, really complex unless you try to think about the phone with a wire. You know those phones? Like the one with a coiled wire that goes to the hearing thing? I never was able to straighten it up. It always goes like all tangled. That's supercoiling. So that's what happens to the DNA molecule. So DNA gyrase straightens it up. So helicase can unzip it. Does that make sense? Topoisomerase 4. It says, breaks down the DNA, okay, to separate two molecules. When there is a replication of bacterial chromosome, does anyone remember what is the shape of bacterial chromosome? Is it circular? Yes, it's circular. So when there's a replication of one circle, you end up with two circles connected like this. This is called concatenated. Okay, that's the fancy word. Concatenated. Okay, connected like this. Try to, okay, like this. Okay? To separate them, topoisomerase 4 introduces the break, pulls them apart, seals the break. Okay? Now, so this is, again, the least that you can go back. Like if you want to make a flashcard or something. Okay? Now we're going to talk about the entire process in more details. Okay? Replication of the DNA is semiconservative. So what it means? This is semiconservative replication. When each of these strands is copied, two new molecules consist of one old and one new strand. Does that make sense? That's as simple as that. Because it was never actually. Now when I tell you, it may sound very obvious that the replication is semi-conservative, but it turns out that we didn't know it, before. We now know it, but it might as well be just conservative, but it turns out this is not true. Okay, this is true. Replication indeed is semi-conservative. Does that make sense? The idea of semi-conservative replication. Okay. That's one foundational concept, okay? Second, it starts at so-called origin. The origin of prokaryotic replication is depicted over here. And if you think about it, let me try to picture it. I'm going to try and picture it. So this is your chromosome. I know it's a lousy picture, but imagine that this is the circular or double-stranded DNA. You see that black thing? Okay. Okay. Now, when you have, that's going to be tough. So this is the replication, and you have two replication forks. Does that make sense? So one fork goes this way, one fork goes this way. Okay, replication goes both directions in a circular chromosome, which makes it very, very, very efficient. This is shown in sort of a better way right here. So I have one fork and another fork. Okay, this fork moves this way, this fork moves this way. Let's name it number 1 and number 2. Okay, does that make sense? So they produce, but it turns out they don't produce 4 molecules. They produce, the result is one and another new chromosome. Each of them consists of one old, which is blue, and one new, which is red, strands. Does that make sense? There's a lot of spatial imagination here. Okay? So, in bacterial chromosome, you're going to find just one origin of replication. Always, just one origin. In eukaryotic chromosome, which is not circular, but is linear, there are multiple origins of replication. In this case, it's easy to imagine like a book. Imagine that you have a very, very, very big book, which consists of many loose sheets. And we have our entire class. And each of us takes a pile of sheets and goes and copies them. Make sense so far? So JCD doesn't copy the entire book, and Michael doesn't copy the entire book. But when we come back together, we can get the new copy of the book together. Does that make sense? Yes?
You're saying the one strand of DNA is like the entire book? It starts copying.
Yes. Yes, let's put it, yes, we can do it this way. Yeah. We can say that one strand of the DNA is entire book. Yes. But the feature is that we have like 2 books which are complementary to each other, right? We have 2 strands of DNA. Does that make sense? Okay. Here is what happens at the replication fork. This is your fork. Okay, let's take a look at each independent protein and figure out what it does. So the first one is helicase. And if you don't mind, I actually would like, it just popped in my mind, if you don't mind, I'm going to write down, since it's all on the video, I'm going to write down the function of the enzymes. So when you rewatch it, you can also see it, okay? So helicase. Anyone remembers what it does? Unzips, yes. Right? Now you have two strands separated. When they are separated, what they immediately want to do, they want to go back together. In order to avoid it, to prevent it, we have another protein here. The one that stabilizes the fork. They bind single strand of DNA. Gyrase is right here, and thank you for reminding me, okay? So single-stranded binding protein stabilizes fork. Does that make sense? As Dominic timely reminded, topoisomerase, what does it do? Prevents supercoiling, yes. It untangles the DNA. So far, does that make sense, those three enzymes? Okay. Now, we need to start thinking how we synthesize the entire thing. Okay? So for this, first we need to make a primer. Very good. Primase. Now we need to introduce the concept. I was trying to put it as far back as possible. The concept of leading and lagging strand. Okay. The DNA polymerase. Here, DNA polymerase 3. It does DNA synthesis. And I will get to the primase, okay? It reads DNA from 5' to 3'. Does that make sense so far? From 5' to 3'. It reads DNA from 5', sorry, from 5' to 3'. Does that make sense? That's this guy, yeah, the brownish thingy. Okay? Okay, and it makes the new strand. Okay, here's the red leading strand. Does that make sense? And if you look at the fork, the entire fork moves in this direction. Does that make sense? So far. Okay? So DNA polymerase can only read from 5' to 3'. Okay? So it can easily read this strand. But if you would look at another strand right here, you can't synthesize something this way, right? It just doesn't read this way. Do you understand why? Think about the book. You read it from left to right. Is that true? I hope so. Imagine that you have another book that is complementary to it, but is written from right to left. Okay? You can't read it this way. You can't. Okay? So it's kind of backwards. Okay? Same happens here, so you have a book written backwards. So what DNA polymerase does, it still reads it from 5' to 3', but it synthesizes, okay, the synthesizes the fragment, then jumps here, synthesizes another fragment, jumps here, synthesizes another fragment. Does that make sense? No? Yes, go ahead. Yes. Oh. I understand the confusion. What I meant is it moves towards the 5' end, and I'm sorry if it made the confusion here. Okay? The leading strand moves towards the 5' end. I should have been more discreet in my explanation. It adds nucleotides to the 3' end of the growing chain. It reads from, well, it moves from 3 towards 5. Okay? The DNA polymerase moves from 3' end to 5' end of the leading strand. Moves. Yeah. I understand. I created the confusion. How to say it better? Moves towards 5'. So well, Slide says it better than I did. From 3 to 5'. It reads from 3 to 5. Okay? Now it starts reading at 3' end towards 5. Yes. Sorry about the confusion. If there was a question, the DNA polymerase 3 reads the old strand of the template strand of the DNA from 3' to 5'. Yes. It grows from 5 to 3, yes. Okay? Now, I'm trying to okay, so we figured that leading strand is continuously replicated. But on the lagging strand, you have DNA polymerase that makes those jumps, okay, and produces what Chesty referred to as Okazaki fragments. Those short fragments of DNA that are synthesized during those backward steps, okay? And this actually allows us to illustrate the role of primers. You see those short green fragments? Those are RNA molecules called primers. The primase right here makes them. Does that make sense? And they mark the beginning of DNA synthesis for DNA polymerase 3. Does that make sense? Every time there's a primer, DNA polymerase will bind the structure, DNA, RNA. Let me clear it up a little. So DNA polymerase will bind the primer and start synthesis. That purple arrow. Okay? Now so you end up with the short fragments of RNA, the primers, and fairly long fragments of DNA, the Okazaki fragments. You need to remove RNA and substitute it with DNA. This is the function of DNA polymerase. RNA removal, DNA fill in. Okay? But nevertheless, when you remove primers, you still have gaps between the Okazaki fragments. Stitching of those gaps is the function of the enzyme called DNA ligase. Does that make sense? More or less. So leading strand is extremely simple. You have a DNA polymerase that moves forward and produces the new DNA strand. But on the lagging strand, you have DNA polymerase, same DNA polymerase 3, that jumps forward, slides back, making DNA, that jumps forward, slides back, making DNA, jumps forward, slides back, making DNA. And those DNA fragments are stitched together by DNA ligase after the primers are removed. Does that make sense? So you end up with two strands of DNA produced in one replication fork, two new strands. Right? So you have two new molecules, each consisting of one old and one new strand. Does that make sense? Okay. Now here is what happens during the chromosomal replication in the prokaryote. You have two replication forks that produce two molecules, circular molecules, which topoisomerase 4 separates at the end of the replication. Like you have two rings, you have to break at least one to separate them. Does that make sense? I want to go back to the previous slide. Okay, here. I'm going to clear it up a little. Now remember, we talked about the leading and lagging strand, right? And the movement of the replication fork. So this there's one mistake in this picture. Can you find it? Yes. Yes. So these two things have to be flipped. Okay? So this should be 3' and this should be 5'. Does that make sense? Okay. Now, here's what happens at the replication fork. At the origin of replication in the prokaryotic cell, you have one complex, one replication fork that moves over here. So this strand becomes leading, right? And this one becomes lagging, because DNA polymerase has to jump and synthesize back. But if you go to another fork, then roles of the strands completely change. Now this strand becomes leading, and this one becomes lagging, because the direction to which fork moves is different, is opposite. Does that make sense? The difference, yes.
So it starts off as a lagging strand.
Say again.
So it starts off as a lagging strand.
What starts off as a lagging strand?
When it starts copying it starts off as a lagging strand.
Think about this. You have four molecules of DNA polymerase 3. Here, 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 and here. 4 molecules of DNA polymerase 3 that make copies. They all start simultaneously, the same origin of replication. Depending on which direction the fork moves, either one or another strand of DNA will be leading or lagging. This is what I mean by that. When the fork moves this way, this is the leading strand. This is the lagging strand. When fork moves the opposite way, then this becomes the leading strand and this becomes the lagging strand. Does that make sense? Okay. That's what I meant when, in the very beginning of this chapter, when we talked about this, I said there's going to be a lot of abstract concept. That's probably the most abstract concept of all. Okay, try to process it. Okay, understand the difference between leading and lagging. Leading is simple. It's continuous replication. The lagging, you have DNA polymerase 3 that hops forward and slides back, making Okazaki fragments. Right, which then, those Okazaki fragments are stitched back together by the DNA ligase. But before that stitching, DNA polymerase 1 has to remove primers, substitute, destroy primers and fill in the gaps with the actual DNA sequence. Okay? Yes? Yeah. So leading strand technically never stops. No. RNA primase here. The primase. It binds with a single-stranded DNA and makes primers, complementary primers. Let me try and show what's going on. I think I have just enough colors. I just need some white space. So this is the DNA. Does that make sense? That's DNA strand. Green is the complementary primer. I'm not going to picture any enzymes. Red is the new DNA molecule. Does that make sense? Now, you see you have new DNA molecule and the primer, which consists of RNA. You need to remove the primer and fill it in with the DNA. Does that make sense? Does that make sense? Over there on the left. Does that make sense? Okay. Now, we will multiply it a little bit. You have 2 primers. You have 2 fragments of DNA. You remove primers. You fill up the DNA ones. Okay. You see the gap? That's the job for ligase. To stitch the gap. Does that make sense? All this complexity happens on the lagging strand. Leading strand is very simple. There is a primer, just one, in the very beginning. There's just one primer. It's not shown here. Okay? This primer would be somewhere like right here. At the origin of replication. Does that make sense? You can see it here, the primer. And then it just goes and goes and goes. Does that make sense? You sure? Think about it. Okay. DNA replication, I must tell you, is extremely reliable. There are almost no mistakes made by DNA polymerase. I don't remember the exact numbers, but we talk about like 1 per million nucleotides, which is very few. If you think especially about the size of the genome of bacteria, which is like maybe 5-6 million nucleotides, every time it gets copied, there are 5 or 6 mistakes, which is not really a lot, okay? And you have mechanism to correct for those mistakes, which we're going to talk about. But first, we need to chat about the mutations. So mutation has a pretty strict definition. Mutation is the change in the DNA or RNA. That mutation leads or does not lead to the alterations in the proteins. But what is important, if somebody tells you mutated protein, the statement is wrong. We all understand what the person means, okay? But mutation, due to its nature, can happen only in the DNA or RNA. When one nucleotide, the proper nucleotide, gets substituted by another. Okay? So, mutations can be a product of mistake during the replication, right? Or during the transcription. Does that make sense? When DNA is transcribed into RNA, mistakes possible, right? So that's when mutation can happen. Most mutations are induced by various external factors. Do you know of any external factors that can cause mutations? What exactly? What type of impact from environment? Yes, you can do temperature mutations, yes. What we are exposed to on an everyday basis. Sunlight, yes, UV light. Something else. Well, generally speaking, yes. High energy radiation like gamma or X-ray. So how would you call it in general? Pollution, chemicals, right? Chemicals. So those are, I would say that radiation and chemicals are two major causes of mutations, two major mutagens. There are of course other ways, but those are two major ones. Okay? So mutations, we're gonna talk a little bit about point mutations. We change one nucleotide in the sequence. So those point mutations can be silent, for example. You see, when there is a substitution in the DNA sequence, which does not change the amino acid encoded. Does that make sense? Like when you substitute in a text, when you substitute large to huge, it's essentially the same, right? Mutations can be missense. When substitution in amino acid leads to the substitution in the DNA sequence leads to the change in amino acid. Does that make sense? So missense means that this changes the protein structure. In most cases, missense mutations make the protein completely dysfunctional. Okay? And you can see that there is, do they have, no, they don't show the proper pairing, but those mutations are usually taken care of, but sometimes they can get into the sex cells and held in ongoing generations. One of the examples from human life, not about mutations, but example with a mistake, in the Bible, there's a story about something like that rich person has, sorry, it's not verbatim, lower chances to get in heaven than the camel to go through the hole in the needle. No, huh? The eye of the needle. Yes, thank you very much. There is a statement, okay? And now think about this: camel going through the eye of the needle. That's kind of a great fantasy to try and to pull the camel in the first place. It turns out, initially in the early versions of Bible, it wasn't the camel; it was a rope. Not the thread, but the rope. Okay? Rope and camel, in whatever language it was, Aramaic or the Greek or whatever, those words were written in a very similar manner, like letter-wise, like maybe one or two letters different. When people copied Bible by hand, they made a little mistake. So rope turned into a camel, and now we know this version. But it was discovered maybe a couple decades ago that it wasn't a camel in the first place. So that's kind of example of mutation that you can see in the text. Same can happen in a human genome or bacterial genome. It may end up with the different protein structure that can either render protein dysfunctional, or maybe protein will gain some new function. For instance, if it's some sort of an enzyme in the bacteria that is inhibited by antimicrobial drug, this enzyme after the mutation may become resistant to the drug. Okay? Nonsense mutation. When instead of the amino acid, stop codon is inserted. So what's going to happen to the molecule of the protein? It's going to be incomplete. It's going to become shorter. And that usually ends up in dysfunctional protein. Does that make sense? Now, insertion or deletion mutations. That's pretty interesting. So this is the deletion mutation here. So I have two amino acids, T and A, okay, that are removed from here. And you can see that this removal completely messes up the triplets. Look at the triplets in here. So one triplet, another triplet, another triplet, and another triplet. When you remove a couple of letters, you have a triplet, then you have something completely, totally different. Totally different. Totally different. Does that make sense? Now this is called frame shift mutation, because you have a certain frame. During the gene expression, the ribosome, when it translates mRNA, it reads triplet codons. It reads using sort of a frame. Does that make sense? When you remove a couple of letters or one letter, you shift the frame. You see what I'm trying to say here? This is why it's called frame shift. But there is a type of insertion or deletion mutations that do not cause the frame shift. Can you think of any? You insert or delete one or two letters, and the frame shifts. Yes.
You have to insert or delete 3 that share the same group of amino acids when they translate.
You just delete any 3. You delete 1 frame pretty much. Okay, does that make sense? And there is no frame shift. You delete 2nd codon or the 3rd or the 4th, and there is no frame shift. Does that make sense? When you specifically delete the codons, there's going to be no frame shift. You're going to cut the piece of the protein out, but that's a different story. The frame will be the same. Okay? Does that make sense? Right? Now this is the example of the chemical that causes the mutation. Nitrous acid, okay, causes changes that are illustrated here. And the C is changed to uracil, okay. The pair GC becomes the pair AT, okay. Does that make sense? And A to hypoxanthine, which actually causes the GC pair to appear. So it causes the alterations in the DNA. That's just an example, the chemical example. Now, does anyone know where you can encounter nitrous acid in a day-to-day life? What it is used for? No? Not really? Processed meats. It is used to keep the flavor and the color in things like salami and bacon. So it's a pretty strong mutagen that we consume on a regular basis if we eat processed meats. And recently there was a study in Britain that linked the consumption of processed meats like bacon and salami and jerky and stuff like that to the increased risk of colon cancer. Yeah, I felt really bad about it. Okay, we're gonna take a break, and when we'll come back,